Thanks so much for the invitation. Amazing program here. My talk will be not only about mitochondrial transplant, but how to better preserve the mitochondria in the organ with simple strategies to make organ preservation better. So I have some conflicts of interest to disclose, which are here. Some of them are related to this presentation in some way. So just to put things in context, 1983 was the first successful lung transplant performed ever in the world, which was done in this hospital here in Toronto, led by this team here, Dr. Cooper and Pearson. And I like to show that because the way we preserve organs since that time it hasn't changed until very recently. And you'll see the mitochondria has anything to do with that change. But basically, the gold standard way was you flush the blood out of the organ in the donor, and you put the organ in a cold bath in a cooler of ice from Canadian Tire. That costs about $20 full of ice, and then you just transport to the recipient center. And I'll show you later it's a bad idea to do that, and I'll explain why. One of the problems with using these strategies is that we had to always rush, right? So you have only about 6 hours from the time the donor is taking from the organ, from the donor to being implanting the recipient, and if you try to increase those numbers of hours, to 8 or 10 hours, the recipients of those organs would pay a price with worse outcomes. So of course there will be a lot of implications if we could extend the organ preservation time, such as overcoming geographical hurdles. We could add better immunological matching. And of course, the ultimate goal of having scheduled transplantation, not like an urgent procedure. And one of the striking aspects of that is when we look at how to preserve these organs. There is old studies, again, from here in Toronto in the beginnings, where all these studies were being done in the lab for preservation of lungs, and at that point was already shown that actually the best temperature of lung preservation was 10 degrees and not ice preservation. Ice preservation keeps between 0 and 4, right? And it basically, the last publication was 1992 that explored this feature. And then this is a Japanese study showing that the intersect was actually 9 degrees Celsius, which showed the best lung preservation. But this was never adopted, and one of the main reasons is that people really didn't know why this was better, right? They just say, oh, it's better, but ice is very practical, let's just continue with ice. And then there was basically 30 years of silence on this topic. And then around the year of 2020, during COVID times, Dr. Ali was a PhD student in the lab, and we started exploring this. And he was, together with our lab, considered the founders of the new era of 10-degree preservation. And basically, his PhD thesis showed that if you keep lungs at 10 degrees, you have much better function, as was shown in the late 80s. But now we know why that happens is because your mitochondria is much better preserved at that temperature. So in his studies, basically he randomized pig lungs to either be preserved on ice or in a refrigerator at 10 degrees, and then those lungs were evaluated after, and we actually did 36 hours of preservation, something that was not imaginable at that time. And we could see that those lungs preserved at 10 degrees, they had very good lung function. Again, I know you're not a pulmonary specialist here, but basically you can see here the static compliance is how easy the lungs inflate and deflate. That's a very good sign of lung healthiness. And you can see that the lungs preserved at 10 degrees had much better compliance compared to the ice-preserved lungs. And we found out that this, again, was a lot related with better mitochondrial preservation. So when you looked at circulating cell-free mitochondrial DNA was much lower. As a consequence, you had much less inflammasome activation and IL-1B production, and also IL-8 and neutrophil activation, which are key hallmarks of ischemic reperfusion injury. Very interesting, we also showed that some of the mitochondria metabolites were active during 10-degree preservation, but not when you keep organs on ice. And one of them is itaconate, which is a relatively new immunometabolite that is produced in the mitochondria and has a variety of anti-inflammatory and anti-cell death properties. We also saw there was less necroptotic cell death. I mean, this is very interesting because we have cell culture studies showing when you preserve cells on ice after 24 hours, you have 49% of necrosis of those cells. If you preserve those cells at 10 degrees, you have only 9% necrosis. So it's a huge, huge difference there too. Now, we moved on to do further studies in transplant models, and we first injured the organs, caused some inflammation, and then we preserved them. So in this case, pig lungs were subjected to aspiration injury, gastric juice aspiration. And then we see that very often in donors that we use for transplant. There's some degree of aspiration. So try to simulate the clinical scenario. And then the lungs are basically randomized to either stay on 10 degrees, go directly to transplant, or stay on ice. And very interesting, we showed here that lungs preserved at 10 degrees had much better function. What was also very interesting here was that they actually had better function than lungs that were directly transplanting. And so if you tell a transplant surgeon and say, please prolong your ischemic time that your organ will be better, they will say you're crazy. But actually, if you prolong your ischemic time at the right temperature, you have these metabolites that are produced in the organ that actually help the organ to heal during that preservation time. And as you can see here, the lung stallage much better preserved at 10 degrees. Now, we did further studies, Dr. Yamanashi from Japan, in LPS, injured lungs. And again, we saw the same thing, and here we explore, mitochondrial membrane potential was much better maintained. You can see here the differential between the red and green, which is the high charge and low charge, was much higher in the 10 degree compared to ice. And then Dr. Wang did a fascinating study looking at metabolomics of these lung tissues. And, when you think about, 10 degrees is actually the temperature of hibernation of mammals. They alternate between 10 degrees and warm temperatures, and then they go back to 10 degrees. Whereas in ice, you can keep something only for a few hours and it deteriorates. So really explain why perhaps we can keep these organs for much longer. And you can see clearly here that there is a huge difference on metabolites, dynamics. At 10 degrees, things are happening in the cells, whereas at ice preservation, things are completely shut down, right? And what is happening? The main thing that is happening is actually the consumption of fatty acids during these periods of ischemia. If you think about, in humans, when we're fasting, we use more beta oxidation, and it's very important. The organ is on ischemia, is also in a kind of a fasting mode. It does use a lot of fatty acids through the carnitine shuttle and for long fatty acids, and direct entrance for the short fatty acids. So you can see here in the 10 degrees, all the fatty acids go down, but on ice, they stay stable. So when you're on ice, you're completely dependent on glucose to maintain cell viability, whereas on 10 degrees, you can use the fatty acids. And see, this seems to be a very predominant mechanism here. related to the mitochondria. And again, complex 1 and 2 are much more active during this time. So, after doing all these studies, we moved on to clinical trials. This was a clinical trial we did in lung transplant that basically we prohibited the surgeons to do any transplant in the middle of the night. So basically the lung will get here, they had to keep the lungs in the 10-degree fridge, and they could only start the transplant from 6 a.m. So of course they all loved this. They don't have to wake up. But as a consequence, the preservation times were much longer, right? And we showed through this study that was actually safe to do that. And you can see here with double preservation times in the 10-degree group, 14 hours versus 8, you had excellent patient outcomes and no difference between the two groups. And then here you can see the patient outcomes, again, from the intervention group versus control. So with growing evidence of this, basically we start moving to 24 hours. And this is a study we did together with a few other centers in Vienna and Madrid. And we can show that up to 200 patients, the lung preservation that used to be only 6 hours, now we're doing about 16, 18 hours, but even going up to 24. And even when you go more than 18 hours of preservation, you do have the same outcomes as with short preservation. This was not randomized data, so we actually did a randomized trial with 310 patients, all these centers here. This was a CHR-sponsored grant. We used this, we developed this, because we couldn't transport a whole fridge to the donor site, so we developed this cooler that actually keep the temperature at 10 degrees. This trial was just finished. We're presenting this month here at the International Society of Heart and Lung Transplant. So the big picture, thanks to the mitochondria, preservation time is not any longer a limiting factor for travel distances. We go to California, we go to Alaska to take lungs today. Daytime transplant for all cases, schedule your implantation time. And the other important factor here is actually economic as well, because we have to send always a private jet to pick up organs because of the rushing time. Now we can use more commercial flights. So a private jet to pick up an organ costs about $40,000 per time. If you go on a commercial flight, it costs you about $1,000. If you go to Vancouver, even if you send your fellows on business class, it will be much cheaper than that. And also you have much less carbon, CO2 footprint because you're not sending jets just for that, and you can do more land transportation. So, from London, Ontario, or Kingston, we used to always take a jet. Now we'll take a car to go there, just again to show some of the impact. Now, this has not been restricted. It started in lung, but now there are many organs that are investigating that, and especially heart, that is a very rich organ in mitochondria, is showing striking benefits also of this temperature. So I'd like to show what is one of the other main reasons why we don't use an ice cooler from Canadian Tire.
The beer is here.
So it's both related to the mitochondria and not to have this type of problem. Now, just to finalize, I showed you how to endogenously modify the mitochondria. There are other, options to do that with some drugs and so on. But also there is a way to have the exogenous mitochondrial replacement, which basically turns to the mitochondrial transfer transplant. And we're also working on that. One of the main advantages today is that we have these devices now. You can see here, this is a normothermic ex vivo lung device. So this is a human lung that is ventilating and keeping at physiological temperature. So this is at 37 degrees. We have used over 1,000 times this machine in clinical cases here for assessment and treatment of human organs. But then you could imagine injecting the mitochondria in this circuit when there is ongoing metabolism at 37 degrees to help heal some of the injured organs. About 80% of all the lungs that are offered for transplantation, they cannot be used today because they have too much injuries. So if you can recover some of those 80% by treating them with mitochondrial transplant, and Dr. Cypel, who is here as well, PhD in our lab, he basically did these studies in pig lungs where he caused a significant injury in the lungs by warm ischemia, and then he took the lungs out and randomized them to ex vivo perfusion with or without mitochondrial transplant. This was mitochondria from pigs to pigs, extracted from pig hearts, expanded, and then injected into the circuit. And what you can see here, again, remember I talked to you about the compliance of the lung is how easy the lung expand, inflate, and deflate. And you can see here when you treat those very injured organs with mitochondria during ex vivo lung perfusion, they perform a lot better with better compliance. They have a lot less edema, as you can see here by the change in the lung weight during perfusion. And they have less pro-inflammatory cytokines as well released during the time of perfusion. So very encouraging data. And we also showed that if you inactivate the mitochondria, using some of inactivation protocols, then you lose the benefit. So, I mean, this is a little bit of a controversial. There are some other studies that shown that even when you inactivate mitochondria, you still get some benefit. At least in our model, if you inactivate them, then you don't get the benefit. So I'll finish here, and thanks so much for the opportunity.